Hello students, now we will discuss few points regarding the differences of protostomes and deuterostomes. These two types of organisms are differentiated majorly on the type of cleavage one and also the conversion of blastopore, which we see in the embryonic development of the organism, into mouth or into anus. So we will see first point, protostomes are the organisms that convert blastopore into mouth. In the same way, in case of deuterostomes, deuterostomes are organisms in which blastopore is converted into anus. Now, what is this blastopore? When it is seen, blastopore is seen during embryonic development. Of either invertebrates or vertebrate type of organisms, of animals. This from what type of animals? That is from porifera to, in case of protostomes, it is up to echinodermata. In case of deuterostomes, again it is up to chordata. To chordata in general. Now, either in case of here or here, the meaning of blastopore is seen. So, blastopore is seen during embryonic development. So, if we go for diagrammatic representation to explain about this blastopore, first of all, we see single-celled zygote. It is getting after mitosis or cleavage into two-celled stage. This one will be like this. So, this is again after mitosis or cleavage, it is getting converted into four-celled stage. Again we see 4 number of cells. It is again converted into, after mitosis, into 8-celled stage. So here we see 1, 2, 3, 4, 5, 6, 7, 8. So again it is converted into 16-cell, later into 32-cell. So in every stage we see mitosis, or we can also use the term like cleavage. So here 16, here 32, later will be 100 number of cells, etc. So during this mitotic division, how the mitosis is happening? In case of protostomes, so in protostomes, the type of cleavage is spiral cleavage or oblique cleavage. In case of deuterostomes, radial cleavage or indeterminate cleavage. We can also use a term like or perpendicular cleavage to the previous type of cells, or perpendicular cleavage. Here we have used the term indeterminate. In case of protostomes, we can also use the term like determinate cleavage. So when the cells are means increasing their number like this, after 32 cell stage, here will be the structure called as blastula. So blastula will be having approximately nearly 100 number of cells in it. Next stage will be gastrula. Blastula, B-L-A-S-T-U-L-A, blastula. Next stage will be gastrula. Now, there will be some shape to the organism. Now, here in this stage, it shows one of the labeling like blastopore. So, how the diagrammatic representation of a blastopore is going to be seen in case of protostomes and how it is going to be seen in case of deuterostomes. That we will see. In case of protostomes, we know that if this is the gastrula stage, and so in case of protostomes, if this is the gastrula, in case of deuterostomes, if this is the gastrula stage, here will be the ectoderm and here will be the endoderm. In case of deuterostomes also, ectoderm and here will be the endoderm. There will be entry. So one of the structure, that is blastopore. Here, if this is the entry, this structure is called as blastopore. So here will be mesodermal tissue. So if the structure which is giving arise to entry of the gut is getting converted into mouth, blastopore into mouth. This opening which is entry of the gut is called blastopore. In course of evolution, when organism is developing, then it has to get converted into mouth. Such organisms are called as protostomes. Now here blastopore, which is entry of the gut, is converted to anus. Such type of organisms are called as deuterostomes. Now, this structure we are giving a term here as archenteron. This is called as archenteron. This is normally we used to call as gut. Now here about this cleavage. So how all these structures are formed, it varies. How here blastopore is getting converted into mouth and blastopore into anus. It all depends upon the types of cleavages. In case of protostomes, if this is the zygote initially, next it will show special type of constriction which is smaller at one area and bigger one. So this will be one cell, this will be another cell. So now we have got two different shaped cells in case of spiral cleavage. Next, again it will undergo oblique cleavage like this. The next stage we will get two small cells and two big cells in the four-celled stage. So this is zygote, one cell. This is two cell. This is four cell. Next, in the 8-cell stage also, we get 4 small-sized cells and 4 big-sized cells. Like this. So this is considered as the oblique cleavage. In case of radial cleavage, if zygote is present, if it has to get divided, then it will give perpendicular every time. So now Imagine that it has undergone one horizontal division like this. So it will give 2 number of cells. In the next stage, it will give perpendicular, means vertical type of division. So now the division has to be like this. So now we will get 1, 2, 3, 4. So again in the next stage, again horizontal division. So we get 8 number of cells. So every time to the previous one, the division is perpendicular to the previously formed cells. So here it is 2 cell, it is 4 cell, it is 8 number of cells. 1, 2, 3, 4, 5, 6, 7, 8. So in this way, the spiral cleavage is seen in case of protostomes, and in case of radial cleavage is seen in case of deuterostomes. This is a small explanation because in the previous MCQs, year MCQs, we have seen the type of cleavages are seen either in protostomes or deuterostomes. Answer will be spiral cleavage or determinate cleavage or oblique cleavage is seen in case of protostomes, and radial cleavage or indeterminate cleavage or perpendicular cleavage is seen in case of deuterostomes.